Hi, my name is Nick Brookhouser, and I'm a graduate student at Arizona State University and an International Foundation for Ethical Research Fellow. My research focuses on the development of a human-induced pluripotent stem cell model of Alzheimer's disease to study the effects of ApoE variants on disease onset and progression. ApoE has been identified as one of the most potent genetic risk factors for late-onset sporadic Alzheimer's disease. However, the mechanism by which this risk is conferred is still poorly understood. Alzheimer's disease, as you might know, is a devastating neurodegenerative disease that affects approximately 5.7 million Americans and is currently the sixth leading cause of death. The estimated cost of Alzheimer's and other dementias in the United States is around $277 billion and could reach over $1 trillion by the year 2050. Animal models have traditionally been used to study Alzheimer's disease and have been proven to be a valuable tool to study the pathophysiology of early onset familial Alzheimer's disease. However, they do not accurately represent the complex genetics of late-onset sporadic Alzheimer's disease. To this end, numerous drug candidates have failed in clinical trials, in part due to our over-reliance on animal models for preclinical development. Currently, there are no effective disease-modifying treatments for Alzheimer's disease, and all current approved drugs are simply palliative. My project involves the development of an accessible and scalable system to generate disease-relevant neural subtypes from human-induced pluripotent stem cells that can be used to study genetic contributions to disease onset and progression. Additionally, using gene editing techniques, we have generated diverse isogenic pluripotent stem cell lines from Alzheimer's disease and non-demented control patients that differ only with respect to ApoE. Isogenic cell lines are a valuable tool in that they vary only with respect to a single genetic location, eliminating the differences in genetic background that may confound results. Together, this provides a controlled system to study the contribution of ApoE variants to disease-relevant pathological hallmarks. Overall, studying disease progression in a human pluripotent stem cell-based system serves as an attractive alternative to animal models for academic investigators to interrogate disease mechanisms, as well as a platform for development and screening of potential therapeutic compounds. This approach will accelerate our understanding of disease-modifying pathways, which is critical for the development of more effective treatments for Alzheimer's disease.